Now I'm going to show how do I obtain the DNA sequences and the phylogenetic tree for our group. And to obtain the DNA sequences, first we need to choose the align and build the alignment for it. For the selection, we create a new alignment and we press OK. And we choose the DNA. The first thing first that we need to do is we need to delete the sequences 1. And after that we need to open our data. And for our group, for our group, the data that we chose is the dataset 1. We open it, and we will obtain the DNA sequences for our dataset 1. But after that, we need to choose the alignment and align by cluster W. And the reason why we need to do this is that by pressing the alignment and align by cluster W, the most similar sequences or the best alignment score will be lined first, and the alignment that we obtain will be much more accurate and much more better compared to the alignment that we didn't align with the cluster W. After we obtain the data sequences, now I'm going to show how to obtain the phylogenetic tree. I'm going to open a file, the same progress, but instead of choosing align, this time we're going to choose the analyze. And with that, we will obtain the analyzed data for our dataset 1, and after that we need to choose the model to choose the best DNA or protein model for our dataset. And with that, we obtain the best fitted model for our dataset. And in this case, the best model for our dataset 1 is T92, which is a Tamura tree parameter, as shown below here that T92 is Tamura tree parameter. After knowing which model is best suited for us, we can construct our phylogenetic tree. By constructing, we press. As you can show here that there is a model for us to choose. And as we know that the model best suited for our dataset is Tamura tree parameter, so we choose that. And we press OK. And with that, we obtain our phylogenetic tree for our dataset 1. After obtaining the phylogenetic tree, we need to save the image to our computer by save as PNG file, and choose the file for our phylogenetic tree. And with that, our phylogenetic tree is constructed and successfully saved to our computer. Now I'm going to interpret the DNA sequences that I have obtained, and in some cases there's some gap here that we can all delete it. For example, from the species 1 here until the species 21, there's all blank here, we could delete it. But in this case, for example, in the first column here, although the species 16, which is the number 4 species, have a gap here in between this all cytosine, but we couldn't delete the gap here as this gap represents the information that we obtain instead of the gap that we can delete it. And this presents the deletion of the DNA sequencer, which gives it distinct characteristics. And in like the second column here, at the species 17, we can see that although this row here is all guanine, but there's a adenine coming out here. It means that it has undergone mutation over time, and it could not be simply changed, as it also carry the information for a distinct characteristic. And this the same goes for all the species. As we can see, as the DNA sequences going backward, we can see that there's a lot of information carried there, and there's a lot of mutation going on and deletion. For example, in here, although species Z, which is the species 1, and the species 11, they carry the information which represent TDT, but as other than these two species, the other didn't carry any DNA sequences for this one, so they leave a blank. There's a gap here, but we couldn't delete it, as this also represent the information of the DNA sequences that species Z and species 11 carry the information, but the other doesn't. And the same goes for the others. Like here, although all the species doesn't carry any information at this column, but species 7 carry the information here, as only this species carry the information here, as is shown here. They get like the name, timing, and cytosine here. And the same goes for the DNA sequences. Not every gap we could delete it, because not every gap is a gap that is useless, but some gap here carry information and we need to interpret it instead of delete it.
Next, a little bit of introduction of phylogenetic tree. First of all, a phylogenetic tree is a visual representation of the relationship between different organisms, showing the path through evolutionary time from a common ancestor to different descendants. The tree can represent relationships ranging from the entire history of life on Earth down to individuals in a population. It is important to understand the relationship between taxa in a phylogenetic tree to comprehend the evolutionary history and relationship of living organisms. In a rooted tree, the branching indicates evolutionary relationships. The point where a split occurs, called a branch point, represents where a single lineage evolved into a distinct new one. Knots are the points at the end of the branches in a phylogenetic tree, representing sequences or hypothetical sequences at various points in evolutionary history, and may be no date. The leaf of a tree, also called tips, can be species, populations, individuals, or even genes. If the tips represent a formally named group, they are called taxa, singularly known as taxon. There are two types of phylogenetic trees. Cladogram displays only the branching pattern of evolutionary relationship among organisms. Next, phylogram represents the evolutionary relationship among organisms by showing both the branching pattern and the amount of evolutionary divergence. In short, cladogram shows related characteristics, meanwhile phylogram shows evolutionary and genetic distance. This is the phylogenetic tree of data set 1. We have scale bar of 0.01. Scale bar is the number of substitution per site, which means the longer the distance, the higher the sequence divergence. From the tree, we can see that species 7 and species 8 are sister species because they share the common ancestor. Species 7, species 8, species 19, and species 6 are the major clade because they share the same knots. Species 9 shows no evolutionary happens. Species 4 and species 13 are also sister species because they are equally related and they have the same length. Species 2 and species 1 are also sister species, but they are not equally related as they have different length. Species 4, species 13, species 2, and species 1 are the major clade. Species 18, species 12, species 13, and species 11 shows no evolutionary happens and they are not equally related. And I would like to emphasize they have different length. And species 7, species 17 and also species 16 shows they might be twin, and they're also sister species because they share the same knot. Species 10, species 20, species 15, species 14 and also species 5, they are major clade. Species 15, species 14 and also species 5, they are the subclade of the major clade, and they share the same length, and which means they are equally related. Species Z is the outgroup because it is more distantly related to the set of species.
Next, which taxa indicates the most genetic changes? That would be sp8. sp8 appears to be the taxon that shows most genetic changes, as sp8 located at the longest branch from the root and compared to its sister taxa sp7. According to Futuyama and Kirkpatrick in 2017, a longer branch for sp8 would indicate it has accumulated more genetic mutation or changes compared to taxa with shorter branch. Secondly, by looking into sp8's sister taxon, which is sp7, the branch leading to sp8 significantly longer than sp7. According to Hill and Mortis, a longer branch meaning more evolutionary accumulate happens.
For question 1.3, which taxa are closely related? Based on the phylogenetic tree, there are few species that are closely related to each other. In phylogenetic tree, the relatedness of two species if they have more recent common ancestor and less related if they have less recent common ancestor. The closely related taxa are species 7 and species 8, which share a very common ancestor, species 4 and species 13, which directly related, species 2 and species 1, which is sister taxa, species 16 and species 17, which are closely related as they share the same ancestor, species 10 and species 20, which are sister taxa, and last but not least, species 15 and species 14, which are sister taxa.
1.4. Which taxa is the outgroup, and why? The outgroup in this phylogenetic tree is species Z. This is because an outgroup is one or more taxa that are known to have diverged before the rest of the taxa in the phylogenetic tree. When choosing an outgroup, it is best to choose species that are less closely related to all the other species we are studying, which is our group of interest, then those species are to one another. If the outgroup is too distantly related, it can be unreliable as it may be difficult to align reliably or have become saturated with substitutions. Determining an outgroup is crucial as it helps to root the tree. The root of a phylogenetic tree is crucial in evolutionary interpretation of the tree because an unrooted tree species shows only the relationships among the taxa and does not define the evolutionary path.